Good morning, everyone. My name is Abigail, and I'm a PhD student at University of Sherbrooke. Today, I will take you through a splice-aware approach to predict genes in eukaryotes. Unfortunately, due to some errors in my implementation, my results have become invalid, and I decided to remove them from this presentation. However, I will explain why this research matters, describe our method, and discuss expected results once the errors are fixed. Despite the small setback, I believe we will still have an interesting talk. A gene prediction is the process of finding coding gene in a DNA sequence. So finding eukaryotic coding gene is difficult because of a very long intergenic region and gene structure that alternates exons and introns. So it's challenging because it requires the identification of multiple boundaries, not just gene starts and ends. And it's like looking for a needle in a haystack. So our research aims to predict transcripts and gene structures by integrating information on splicing into our model. So we can classify current gene prediction methods by answering this simple question. Is a gene structure model imposed from the start? If the answer is yes, it's a model-based approach. And if it is no, it's a model-free approach. So despite all gene prediction tools developed, some problems like alternative splicing remain unresolved. Most computational tools predict a transcript for each gene instead of predicting genes by considering alternative splicing. So given this limitation, the prospects for improving gene predictions methods lie in developing algorithms that consider alternative splicing reality. So we propose a novel model-based gene prediction approach. And the first step of our method is design an HMM model technique that integrates alternative splicing information. So this is our HMM model. Blue denotes transcript states and yellow represent alternative states that we added to integrate splicing information. Errors are transition between states. So step 2 of our method is data retrieval. So we use human genomic data for chromosomes 1 to 22 in the Ensemble 98 database containing 9,504 forward genes and 33,780 transcripts. So 80% of the data is used to train our model. So to validate our approach, we will test our model on 20% of the database and we'll evaluate its accuracy in predicting gene structure using standard metrics like precision and recall. So step 3 is transcript annotation. So we annotate transcripts within our data set using states like start codon, exons, donor sites, introns, acceptor site, and stop codon. Step 4 is gene annotation. So following transcript annotation, we deduce the corresponding gene annotation. If a particular state does not appear across all transcripts, it is considered an alternative state within the gene. So here is an illustration of how we annotate a gene based on our model. So this gene has 3 transcripts, transcript 1, transcript 2, and transcript 3. The transcript 2 is the first one to start. He starts with a start codon, followed by an exon. So we annotate the gene the same with the same codon, start codon and exon, because it is the only one and at that position. Afterwards, we see that transcript 1 and transcript 3 have started in this position with start codons. So at this position, we have start codon for transcript 1, transcript 3, and exon in transcript 2. So in the gene, we will annotate alternative start codon, just to say that there is a start codon, but who doesn't appear across all the transcripts. So it's an alternative one. When we move to the next position, we can see that transcript 1 and 3 are exons, while transcript 2 is a donor site. So we annotate the gene as at this position as an alternative donor site, because this donor site does not appear across all transcripts. So the same thing for the next position. Transcript 1 and transcript 3 are exons, while transcript 2 is an intron. So we call that state in the gene X intron, to say that there are some transcripts which are exons and other one introns on that position. And when we move again to the following one, and we see that transcript 1 and 3 are still in exon state, while there is an acceptor site in transcript 2. And we annotate the gene as at this position as an alternative acceptor site, because there is one that is not across all the transcripts. And at this position, all the transcripts have exons, so it will be exons in the gene. And the following one, only one transcripts have a donor site, the other one are still exon. So we will notify that there is an alternative donor site at this position in the gene. And we have the same thing here, exon introns means exon in the gene. Here we have an acceptor site, exon, so it's an alternative acceptor site in the gene. And here we have the transcript 3 stops here, so it has a stop codon, while transcript 1 and 2 are still exons. So we annotate it like an alternative codon, stop codon. And the transcript 3 has finished here, so now we just have transcript 1 and 2. There are exons in this position, so it's an exon in the gene, and they finish by stop codon. So this is the complete workflow of our method. It helps us to predict gene sequences, gene and transcript sequences. So upon error rectification, our model is expected to provide increased accuracy on gene and transcript annotation. And with more accurate prediction of gene and transcript structure, our method will lead to precise annotation of the genome. And as a future application, we will look at transcript and gene evolution. Our method provides detailed insight into gene structure and transcript that will allow for in-depth analysis of gene and transcript evolution. So what is the road ahead? In the short term, our focus is on refining our model by fixing program errors and applying into actual genomic data. So this step will validate the model efficiency and its ability to make precise gene and transcript predictions. Our medium-term objective involve extending the scope of our model. We aim to include the entire structure of a gene, like 5' UTR and 3' UTR regions. In the long run, we intend to use our model for genome annotation. By considering alternative splicing, we aspire to produce more detailed and accurate annotations of eukaryotic genomes. So in conclusion, we intend our model to make big improvements in gene prediction, and we can't wait to see how it changes the field. So I would like to acknowledge the dedicated efforts of our research team, Cobius Lab, and the valuable contribution made by my colleagues. Without their hard work and insight, this project would have not been possible. I gratefully acknowledge the financial support provided by the Canada Researchers and the Natural Sciences and Engineering Research Council of Canada.
Okay.
Special thanks.
Thank you. So, any question? Okay. Yeah, I asked a question. So, do you have a slide to show your HMM model?
Sorry,
The structure of the HMM model.
I can't. Can you repeat the question, please?
Do you have a slide to show your HMM model, the structure of the model?
The framework?
Yeah.
Do you mean?
Do you have the HMM model? You have a graph. Yeah. Okay. The slide 4. Okay, then you have it. It's good.
Is it this one, the HMM model?
Yeah. Okay. That's good one. Sorry, I missed this slide. Thank you.
Okay, thank you.